Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we will be covering Chapter 3 of the Bauman 6th edition textbook, Biology 2420, which covers cell structure and function. So let's go ahead and get started. Recall from Biology 1406 that there are certain fundamental processes to life, and living organisms possess most of these, if not all of these processes. So for example, living things grow, they reproduce, they respond to their environment, they metabolize. And remember that viruses are not alive because they can't do a number of these processes. So let's talk about what life entails. Remember, living things fall under 3 domains of life, one of 3, and that is living things are either in the domain bacteria, the domain archaea, or the domain eukarya. Remember that viruses are not organisms. They do not fall under any of these domains of life, and therefore viruses are not alive. So let's start with prokaryotes. Remember, there are 2 domains of life which consist of prokaryotic organisms. Those are the bacteria and the archaea. These prokaryotes do not have a proper nucleus. They do have DNA. They do have, genomic DNA. They have a chromosome. Usually there's only 1 chromosome. And by the way, here's a fun fact for you. The chromosome of a prokaryote is circularized. What does that mean? It's still double-stranded DNA, but there is no beginning to the DNA strand and there is no end to the DNA strand. So you have circularized double-strand DNA, and prokaryotes usually have a 1 circularized double-strand DNA, and that makes up their genome. 1 chromosome of DNA, whereas you and I, our chromosomes are not a circle. They're linear, right? Linearized, and we have usually multiple chromosomes. Humans have 46 chromosomes, right? Well, prokaryotes usually only have 1 chromosome, and that chromosome is circularized. Fun fact for you. Now, what is the DNA for? Do you remember why we have DNA? Well, because the DNA harbors the genes, and each gene is the information for a protein. That gene information is copied into messenger RNA. That's a process known as transcription, and then that messenger RNA is translated into a protein. Okay, so this happens in prokaryotes, this happens in bacteria, archaea, and actually that process happens in eukaryotes as well. So again, we don't have a proper nucleus. What does nucleus mean? Nucleus means that the genomic DNA is surrounded by a membrane, right? Eukaryotes have a nucleus because the DNA is surrounded by a nuclear envelope, right? It's a membrane around the genomic DNA. Prokaryotes, their genomes are not surrounded by a membrane, so it's not a proper nucleus. Instead, it's called a nucleoid. Let me show you that word here. Let me show you that word real quick. Look here on this slide. This DNA here is called a nucleoid. Nucleoid. See the chromosome? If you were to take this chromosome out and spread it apart, it would be one big double-stranded circle. Okay, but instead you see that big double-stranded circle all clumped together in the center of the cell, or somewhere near the center of the cell, and that's called a nucleoid. All right. Now, what else? Again, these prokaryotes, not only do they lack a nucleus, which means they lack a membrane around their DNA, they also lack any organelles that have a membrane. So they lack intracellular membrane-bound organelles. For example, what are intracellular membrane-bound organelles. Well, have you heard of the chloroplasts? Have you heard of mitochondria? Have you heard of the nucleus? Have you heard of the endoplasmic reticulum? Have you heard of the Golgi apparatus? All of these are examples of membrane-bound organelles. You're not going to find those organelles in a prokaryote. So basically, if it's inside of the cell and it has a membrane around it, you're not usually going to find that in a prokaryote. That's an easy way of thinking about the difference between prokaryotes and eukaryotes. Prokaryotes lack intracellular, intracellular membrane-bound organelles. Okay, and usually because of that, they are very small cells. These prokaryotes are smaller, usually, than 1 micrometer in diameter. What's a micrometer? It's 1,000th of a millimeter. You guys know what a millimeter is, right? 10 to the minus 3 meters. Well, this would be 10 to the minus 6 meters. This is 1,000 times smaller than a millimeter. Okay, and like I said, the bacteria and the archaea make up these prokaryotes. So let's talk about eukaryotes. Eukaryotes, on the other hand, are more complex cells. These are the cells that your body and my body are made of, eukaryotic cells. Eukaryotic cells have a nucleus. That means our chromosomes are surrounded by a envelope, a membrane, if you will. It's actually a double membrane called the nuclear membrane. And then we do have internal membrane-bound organelles. You know, for example, like I said, the nucleus is the main membrane-bound organelle. Then you can have the endoplasmic reticulum, the Golgi apparatus, mitochondria. If you're a plant, which I hope none of you are plants, because that would be strange, but, you know, plants have chloroplast organelles as well, right? And these are larger cells. Obviously, if you're going to have all these membrane-bound organelles, these are going to be some large cells, right? 10 to 100 micrometers in diameter. So they tend to be 10 to 100 times, or even more, 10 to 100 times larger than prokaryotic cells. So eukaryotic cells are much larger than prokaryotic cells, usually between 10 and 100 times larger than a prokaryotic cell. They're more complex. Remember, what do these include? When we're studying microbiology, there are examples of eukaryotic microbes, algae, protozoa, which, by the way, the algae and the protozoa are the protists. These are the protists. Fungi, animals, plants, all of these can be studied as eukaryotes. And also, there are examples of each of these as eukaryotic microorganisms as well. Okay, so look at this. Well, actually, let me show you this first. These are the common features of prokaryotic cells. So what do we have here? Remember, we don't have a proper nucleus, do we? We have a nucleoid, which means that the DNA is not surrounded by a membrane. Remember? And how many chromosomes are in a prokaryotic cell? Do you remember? Yeah, that's right. Usually only one chromosome. And what does that chromosome look like? What does this look like? Well, you're like, but Dr. D, that looks like a bowl of spaghetti, right? But what if I were to take this bowl of spaghetti out and stretch it apart? What would it look like? Remember what I said earlier? That's right. It would be one really big circle. There would be no beginning to the circle and no end to the circle. Does that make sense? In a prokaryotic cell, you usually have one chromosome, which means one circularized, no beginning, no end, double-strand DNA. So you'd have double-strand DNA in a big circle, and that's what the nucleoid is, okay? Does that make sense? And there's no membrane around that double-strand DNA. So that's what is going on inside of most prokaryotic cells. And then what's the fluid in here? What's the fluid in here? What's that? You guys remember from biology 1406 what the fluid is called? That's right, the cytoplasm or cytosol, right? Cytoplasm, cytosol. What are these little peach dots everywhere? You see these little dots everywhere? Small dots? Those are the ribosomes ribosomes. Ribosomes are the organelle. By the way, ribosomes are organelles, but ribosomes are not membrane-bound organelles. Does that make sense? Ribosomes are not membrane-bound organelles. They're just organelles. That's why prokaryotes have them. And remember the function of ribosomes. Ribosomes are there to undergo a process called translation. That means producing proteins. Ribosomes synthesize proteins from genomic DNA, from gene information. All right, what else do we have? Do you see this dark brown membrane here? This dark brown right here? This membrane? That's called the plasma membrane. What you should know is all cells have a plasma membrane. All cells have a plasma membrane. And then what's this out here? This more orange line, or I guess mustard colored line. That's the cell wall. Prokaryotes have cell walls. And do you guys recall that we briefly mentioned in chapter 1 that the cell wall of a bacteria cell is comprised of or composed of peptidoglycan. Whereas the cell wall of archaea is not composed of peptidoglycan. Okay? I want you to know that eukaryotes do not have a cell wall of peptidoglycan. Archaea do not have a cell wall of peptidoglycan. Only bacteria have a cell wall of peptidoglycan. In fact, you should know that peptidoglycan is unique to bacteria. What does that mean? Only bacteria are known to have peptidoglycan cell walls. Okay? And by the way, not all bacteria have a cell wall, but most bacteria have a cell wall. Okay? Well, that's a good overview of a general bacterial or archaeal cell. What is this? See at the top left? This is a typical eukaryotic cell. A typical eukaryotic cell. So what does a typical eukaryotic cell have? Obviously this giant purple structure right here. You see this? That's the nucleus. You see that this is the double membrane of the nuclear envelope. And inside of the nuclear envelope, what do you have? Your chromosomes. If this was a human cell, you'd have 46 chromosomes inside. And remember, human chromosomes or eukaryotic chromosomes are linearized. It's double-stranded linear DNA. Okay? And then outside, you have other membrane-bound organelles. Remember all the membrane-bound organelles we talked about in previous classes, like 1406? This is the rough endoplasmic reticulum. Then you've got the smooth endoplasmic reticulum. You've got the Golgi apparatus. You've got mitochondria. If this was a plant cell, which it's not, but if this was a plant cell, you'd have chloroplasts as well. So all of these are structures that you would not see in a prokaryotic cell. And that's why these cells are so much bigger. See, this is a much larger cell, 10 micron cell, whereas this cell here would be 1 micron or less. Does that make sense? Prokaryotic cells are 10 to 100 times smaller on average than a eukaryotic cell. Okay, in fact, you can see viruses which are not alive. They are small infectious agents made of only protein and nucleic acid. You see, they are 0.3 microns usually, okay, or about 0.3 microns, whereas prokaryotic cells are about 1 micron, and then eukaryotic cells are, more than 10 microns. Does that make sense? So eukaryotic organisms are going to be in the 10 plus micron range. Bacterium and archaea are going to be in the 1 micron range, and viruses are going to be in the 0.3 micron range. By the way, The microscopes we use in lab, the best they're going to resolve is about this. It's about 1 micron. You are not going to see structures that are 0.3 microns with a light microscope. You know, the type of microscope we use in the lab, the compound light microscope. So you cannot study viruses, for example. You can't study viruses in a common lab, in a typical microbiology lab, because our light microscopes cannot resolve at this level. We can study bacterium and we can study eukaryotes, but we can't study viruses using a compound light microscope. And I'll be talking about, in a different chapter, we're going to be talking about how you can visualize viruses using more specialized microscopes, namely electron microscopes. We're going to be talking about that in a subsequent chapter. So, what do these cells look like when you're looking at prokaryotic cells, namely bacterial cells? What do these cells look like? Well, there are many different shapes, but there are 2 most common shapes, okay? I want you to know that bacteria commonly are either coccus shaped, which means spherical shaped, or bacillus shaped, which means rod shaped. And then that refers to shape. I need you to understand that when we're talking about bacteria, there's a difference between talking about bacterial shape versus a bacterial there's shape and conformation, okay? There's shape and arrangement, I should call it, okay? Arrangement. So, if I were to ask you, what's the shape of this bacteria, you would say coccus. And the plural, coccus would be the plural of spherical cells. Cocci is the plural, coccus is the singular. Okay, coccus is the singular, cocci is the plural. Bacillus is the singular, bacilli is the plural. Okay, again, shape, coccus, singular, cocci, plural. So if you have several spheres, you have cocci. If you have a single sphere, that is coccus. If you have a single rod-shaped bacteria, that's bacillus. If you have multiple rod-shaped bacteria, that's bacilli. I know it's a tongue twister. I almost got it backwards myself, because you know it is easy to mix up these terms. But again, coccus singular, cocci plural, bacillus singular, bacilli plural, right? So that has to do with what? That has to do with shape of the bacteria. Now let's talk about arrangement, okay? This is very important. Arrangement. Arrangement means, how are those coccus bacteria? How are those bacillus bacteria? How are they arranged? How are they grouped up, right? So, for example, if you have a chain, and sometimes you see this under a microscope, if you have a chain of bacteria, that's called strepto. Look at this. Here's a chain of rod-shaped bacteria. That's strepto. Here's a chain of spherical bacteria. That's strepto. So if I saw this under the microscope, I would say I have strepto arrangement and cocci shape, right? I have a chain of spherical bacteria. Does that make sense? So I would say this is streptococci because I have the arrangement strepto and I have the shape cocci. Whereas if I saw this under the microscope down here, I would say I have strepto, which means chains of rod-shaped, bacillus, rod-shaped bacteria. Does that make sense? Strepto is arrangement, bacillus is the shape. Strepto is the arrangement, cocci is the shape. All right, I hope that made sense. There's a big difference in microbiology between arrangement and shape. Arrangement means how are they grouped up, and shape means how does each individual look. Okay, and then there's this one here I want you to know about. You don't need to know about Sarciniae. What we should focus on is Staphylo. Staphyla means grape-like cluster. So if you see grape-like clusters, that's Staphyla. And usually you don't see grape-like clusters of rod-shaped cells. So usually this is specific to spherical cells. So if I saw clumping like this looks like grapes, I would call this Staphyla arrangement. And what's each shape? That's right, coccus shape. So I would call this Staphylococci. Okay, does that make sense? Staphylococci looks like this. Streptococci looks like this. Then you can have coccus. You could have, two spherical bacteria side by side. That would be Diplococcus, and so on and so forth. Diplococci would be, like I said, two spherical bacteria side by side. Coccobacilli would mean bacillus that are short, almost like a combination of coccus and bacilli. If you have really short rods, that would be called coccobacilli. If you have two bacillus, that would be diplobacilli. Okay, so, again, there are shapes and arrangements. Please learn both. And then you can have more complex shapes too. We'll talk about later, but you could have Vibrio, which are comma-shaped bacteria. You could have Spirillum, which are spiral-shaped bacterium. There are many, many different shapes to bacteria. All right, so before we go into some of the more common features of bacterial and archaeal cell structure, let's take a quick break time with Tig and come right back. Hey everyone, welcome back from break time. Let's go ahead and keep going. So common features of bacterial and archaeal cell structure. Remember, these are prokaryotic cells, so you're not going to see any membrane-bound organelles. So here's some terminology for you. When I refer to the cell envelope, I'm talking about all the layers that surround a particular bacteria or archaea. So this would include the plasma membrane. Remember, all cells have a plasma membrane, the double phospholipid bilayer around the cell. The cell envelope can include a cell wall. Remember, if we're talking about bacteria, that cell wall is comprised of peptidoglycan, whereas if you're talking about an archaea, there are different structures to the cell wall there. The cell envelope can include a capsule or slime layer, also known as the glycocalyx. This is a gooey structure made of sugars and proteins outside of the cell wall of some bacteria. All right, so again, cell envelope can include a plasma membrane, cell wall, or capsule or slime layer. Not all bacteria have a capsule. Not all bacteria have a slime layer. Not even all bacteria have a cell wall. Remember, I said there were some bacteria that do not have a cell wall. But some bacteria do have these structures, if that makes any sense. And remember what the nucleoid is? It's the circularized genomic material, the double-stranded DNA that is in the shape of a circle, circularized. And usually how many chromosomes are there? Quiz time. How many chromosomes does a prokaryote have? Usually one chromosome. And then remember the ribosomes. The ribosomes are the organelle responsible for protein synthesis, for translation in the cell. Remember, ribosomes do not have a membrane. And inclusions. Inclusions are just areas scattered through the cytoplasm of storage. So you could store various resources for the cell. So let's talk about these structures. Cell envelope. Remember I said all cells have a plasma membrane, but the cell envelope could include a cell wall or one additional layer like a capsule or a slime layer, right? So let's talk about those capsules or slime layers. Remember, those are called glycocalyxes, and these are gelatinous, sticky substances around the cell. They could be composed of polysaccharides, these are sugars, or polypeptides, these are proteins, or both. Okay, so it's this gooey substance outside of the cell wall. And remember, not all bacteria have a glycocalyx. Remember, glycocalyx includes the capsule or the slime layer. All right, so what else are we dealing with? Oh, remember I told you earlier, cells have not just spherical or cocci shape or rod shapes, that's known as bacillus, but remember I said there's other shapes to bacterial cells: vibrio, spirilla, hyphal, pleomorphic. Pleomorphic, by the way, means bacteria that are variable in shape. They don't have a particular shape. So let's look at some of these real quick. Remember, coccus shape is spherical. Here's some coccal bacteria as they appear under the microscope. Rod shape means bacillus. These are bacillus bacteria as they would appear under the light microscope. Spirillum, these are spiral shape. Spirochetes, these are even more spiral shaped. They're longer. An example would be syphilis is a spirochete. And then there are bacteria with appendages like stalks or hyphae. And there are filamentous bacteria with hyphal shape. This is hyphal shaped filamentous bacteria. These bacteria consist of elongated cells. Actinomycetes is an example. We're going to learn about that later in the semester. But you know how mold grows with hyphal shaped, this elongated cell shape? These bacteria grow kind of like mold in that they have elongated cells. And what is the term for a bacteria that does not have a particular cell shape? Do you remember that? That's called pleomorphic. Pleomorphic bacteria do not have a particular cell shape. And you know why they don't have a particular cell shape? It's usually because they don't have a cell wall to keep them in a particular shape, okay? So the bacteria, remember I said not all bacteria have a cell wall. Well, the ones that don't have a cell wall, they're not going to be held into any particular shape. So they're going to be pleomorphic, if that makes any sense. Remember the glycocalyces? We said bacteria, some bacteria have a glycocalyx. You can either have a capsule or a slime layer. This is part of the cell envelope. The capsule is usually firmly attached to the cell surface, and it can be used to prevent the bacteria from being recognized by the host. So imagine you get a bacterial infection, and this particular bacteria that infected your body is surrounded by this gooey stuff, right? This sugary, proteinaceous stuff that's around the cell. Well, that could make it harder for your white blood cells to identify the bacteria as a foreign invader, right? What do your white blood cells do in your body? Remember, white blood cells in your body, they recognize bacteria. They recognize foreign invaders and rid them from your system, right? Well, if the bacteria has a capsule, that can effectively cloak the bacteria from your immune system, right? And make it harder for your immune system to fight off that particular bacteria. So what about slime layers? What you need to know is slime layers are loosely associated with the cell surface. They're loose, whereas capsules are tightly stuck to the cell surface. Slime layers are loosely associated with the cell surface. In fact, they tend to be water soluble. This is a sticky layer that allows the prokaryote to attach to surfaces. Okay, so you can see here the difference, the big difference here. Here's a capsule. Notice how the capsule is so tightly associated with the cell wall. Here's a glycocalyx that's a slime layer. See, the slime layer is very loose, very loosely associated with the cell wall. The capsule is mainly, the function is to evade the host's immune system. The glycocalyx that's a slime layer, the slime layer is used to attach to surfaces. Now, some bacteria even have flagella. Flagella are long whip-like structures that, or extensions that allow for movement, like a propeller. The prokaryotes use these flagella kind of like a whip-like propeller to propel the cell forward. It's a type of motility of the cell, and the flagella has certain parts to it. There's what's known as the filament. This is the long whip-like structure. The hook, which attaches the whip-like structure to the cell, and the basal body, which anchors the flagella to the cell. So you can see here the flagella, you see the flagella is this long whip-like structure. The flagella or filament, the filament is the long whip-like structure, and then the hook attaches that filament to the basal body. The basal body is attached to the cell wall of the bacteria. And the way these flagella work is that due to a proton gradient or proton diffusion of protons across the membrane, that causes rotation of the flagella. The flagella literally spins like a propeller, and that propels the cell through the fluid. Okay, so here's a bunch of bacterial images with a scanning electron microscope. And you can see these long whip-like structures. You see these long whip-like extensions off the cell. Here, this cell only has one. This cell has a tuft. This cell is surrounded by flagella. All of these are possible. This arrangement is called peritrichous. Trichous means the flagellar of arrangement, right? Peritrichous bacteria have flagella all around the perimeter, okay? Whereas monotrichous or polar trichous, polar flagellum, are bacterium with one flagella, monotrichous, one flagella. Or you can have lopotrichous. Lopotrichous means a tuft. So you've got several flagella in one spot. So you can have atrichous. What do you think atrichous means? Atrichous means the bacteria has no flagella. Monotrichous means it has one flagella. Peritrichous means it has flagella all over the place. And lopotrichous, that's L-O-P-O-trichous, means you have a tuft. You have several flagella in one spot, as in the bottom image here. And then do you remember the spirochetes I told you about, the spiral-shaped bacteria? The spirochete bacteria, these have a different kind of motility. They have flagella, but the flagella are internalized. Look at this, it's called endoflagella. Endo means internal flagella. So there are flagella that are inside of the cell wall, okay? And because of that, they form what's known as the axial filament. When the flagella rotate, that causes the whole cell to rotate. Does that make any sense? The flagella are inside of the cell wall, so when they spin, the whole cell spins. The whole bacteria spins. And what's this bacteria shaped like? A corkscrew, right? Like a spiral. And just like a corkscrew can bore its way through a cork of a wine bottle, these spirochetes can bore their way through mucous membranes in your cells, between your cells, I should say. Interesting stuff. Now, more information about flagella. You know, flagella spin like a propeller on a boat. Remember I said the flagella spin like a propeller? Well, it turns out that the direction of the spin is important. If the flagella is spinning counterclockwise, this means that the bacteria will be propelled forward. If it's spinning clockwise, this means that the bacteria will tumble around in place. So let me show you. Here's a bacteria that's tumbling. Tumble means the bacteria is just staying in place, but kind of flipping around, tumbling, like flipping its direction around, but staying in place. The flagella spin clockwise, and this causes tumble. But when the flagella switch to a counterclockwise rotation, this causes what's known as a run. The flagella runs. The flagella moves. The bacteria moves in a straight direction. You see, and then the flagella switch back to clockwise rotation, and there's a tumble, and then switch back to counterclockwise, and then there's a run. So the way bacteria move is kind of like those cheap remote control cars. You ever bought one of those really super cheap remote control cars that only has one button? Okay, I like to explain it this way. Have you ever seen a remote control car with one button? And you say, but Dr. D, how do you drive a car that has one button, right? A remote control car with one button. Well, if you hold that button down, what happens? Your little remote control car goes straight, doesn't it? When you let go of that button, the little remote control car does little donuts. It spins around. You see what I'm saying? So you let go of the button, the remote control car spins around and around. And then once it's pointing the direction you want, you hold the button and it goes straight. Bacteria are kind of like that. When they want to go straight, they make their flagella spin counterclockwise. When they want to tumble or change direction, they spin their flagella clockwise. Very interesting stuff. So if you want to see motility under the microscope, if you want to see a bacteria that's moving around under the microscope, and that bacteria has flagella, you're going to expect to see runs and tumbles. Each bacteria is going to go straight for a while, then kind of vibrate in place and move around and tumble around in place, and then move straight. So that's what you're going to see. Flagellated bacteria move around with runs and tumbles. All right, here's some more extensions or external structures you might see on bacterial cells. These are called Fimbriae and pili. Fimbriae are extensions of the protein extensions that are sticky, bristle-like projections from the cell. These are not there for motility, but they are, because they are much shorter than bacteria, they are there to stick to surfaces. Okay, they are used to adhere to one another or to substances in the environment. They serve to function in biofilm formation. You guys know the most common biofilm that you're probably aware of? How about plaque on your teeth, right? You've got plaque on your teeth sometimes, or a film that appears on your teeth. How did those bacteria stick to your teeth? Well, bacteria can stick to the very smooth surfaces of your teeth because they have Fimbriae. The Fimbriae, along with slime layers as well, remember a type of glycocalyx, the Fimbriae and slime layers allow the bacteria to stick to your teeth, and then the bacteria ooze out this goo that forms into the biofilm, and then other bacteria stick to the biofilm, and now next thing you know, you have a little community of bacteria growing on your teeth, forming what's known as plaque. So here you can see a bacteria. This would be what kind of... Oh, here's a quiz time for you. What is the shape of this bacteria? Shape. That's right, bacillus. It's a rod shape. Is it one bacillus bacteria or two? It looks like two bacteria. So I would call this what? Diplobacillus, right? Diplobacilli, I should say. Diplobacilli. Anyway, so this is two rod-shaped bacteria. Do you see the long extensions here, these long whip-like structures? What do you think that is? That's the flagella, isn't it? So this is a peritrichous. That because there's flagella all over the place, right? Peritrichous bacteria. But what do you think these short extensions are? You see these very short extensions? It looks like grass. That's the Fimbriae or Fimbria. The Fimbriae are responsible for attachment to surfaces, such as the surface of your tooth. Okay, and what's a pili? What's a pili? It's another extension from the cell, but it's got certain functions. And the one I want to talk about is called the conjugation pili. This is a hollow tube that forms from one bacteria that can attach to an adjacent bacteria. And then this allows for transfer of DNA from one cell to another, a process known as conjugation. It's so interesting. Look, here's a bacillus bacteria, right? Here's a bacteria. Look what it's formed. Look at this. This is actually a hollow protein tube, okay? So imagine this bacteria forms what's known as the sex pilus, which is this hollow tube. It extends until it attaches to, let's say, this bacteria. So now this bacteria is now attached with the sex pilus to this bacteria. And do you remember what it said? DNA exchange can happen. Actually, it's a one-way DNA exchange, which means DNA can be shared from this bacteria to this one, not the other way around. So this bacteria can pick up information from this bacteria. This is a process known as conjugation. It is a form of what's known as horizontal gene transfer. What does that mean? Horizontal gene transfer. It's strange. You and I are humans. When we do gene transfer, that means from parent to child, right? That's called vertical gene transfer. If I and my wife were to have a child, that child would inherit genes from us, right? That's called vertical gene transfer. That means from one generation to the next. Does that make sense? But this is not vertical gene transfer. No children are being made here. Remember, because bacteria cannot mate sexually. Bacteria mate asexually. So, you know what that means? That means that this is horizontal gene transfer because no children are being made. This adult bacteria is sharing genes with this adult bacteria. That's what horizontal gene transfer means. Interesting stuff. So I bet you didn't know that bacteria can share genetic information with one another. It's very interesting stuff. And they do it without the process of sexual reproduction. Very interesting. And by the way, why is this scary? Because, you know, there are genes that some bacteria have that make them, let's say, resistant to an antibiotic. Let's say that this bacteria has this important gene called beta-lactamase. Well, that's the gene that makes a bacteria resistant to penicillin. Does that make sense? So let's say this bacteria had the gene that makes the bacteria resistant to penicillin. Well, through conjugation, horizontal gene transfer, this bacteria could very well share that gene with this bacteria. Now, suddenly this bacteria is also resistant to penicillin. Why? Because it got the gene from this other bacteria. You see, so bacteria can share information with one another, and sometimes that information is the information to become resistant to an antibiotic. The information could be to make a toxin. That's not good. Let's say this is a bacteria that can make a toxin. Let's say this is Shigella bacteria, which makes the very dangerous Shiga toxin. Imagine if that Shiga toxin gene was shared with this bacteria. Well, now this bacteria can also make that toxin, and so that bacteria becomes dangerous, doesn't it? So this conjugation can actually lead to a lot of chaos when it comes to healthcare and, you know, what you should be aware of that bacteria that can do. All right. So before I go on any further, let's take one more quick break time with Tig, and then we'll come back and finish off this chapter. All right, everyone, welcome back from break time. Let's keep going with bacterial cell walls. Now, why do bacteria have cell walls? Well, it's a protective structure. It prevents the cell from rupturing due to osmotic forces. Let's say water rushes into the cell and that causes the cell to swell. That can cause lysis of the cell. The cell can break apart. Okay, so bacteria require cell walls, or at least most do, because remember, not all bacteria have a cell wall. Bacteria need cell wall to provide structure, shape, and to protect from osmotic forces. Now, the bacterial cell wall can be the target of many antibiotics. If you've heard of penicillin, for example, penicillin targets the maintenance of the cell wall. So the cell wall is not able to, to do its normal function, to stay intact, and the cell swells and lyses due to osmotic forces. That's the mechanism of action of penicillin. Very interesting stuff. So again, the cell wall protects the cell. It prevents against osmotic forces from destroying the cell. It gives the cell shape, you know, the coccus shape, bacillus shape, spirillus shape. all of these shapes. And remember, in bacteria, the cell wall is composed of peptidoglycan. I'm going to explain in a couple slides what peptidoglycan looks like and how it fits together to form the cell wall. Now, what you need to understand is when you're talking about the bacterial cell wall, there are two types of architecture that you're dealing with. There is what's known as the gram-positive cell wall, where the cell wall looks a particular way, and then there are what known as gram-negative cell wall, and that those bacteria have a cell wall with a different architecture. So let's talk about this. First of all, what is peptidoglycan? This here on the right is a basic building block of peptidoglycan. This is one unit of peptidoglycan. Notice it is a disaccharide, which means two sugars. Here's a sugar, here's a sugar. Just to compare, this is a glucose molecule, so very similar to a glucose molecule. You've got a sugar here and a sugar here. Now, not only is peptidoglycan made of these two sugars, but also amino acids that are attached. You see, these are proteins that are attached. It's called a peptide chain, which is attached to the NAM, basically, to N-acylmuramic acid. NAM has a peptide chain attached. So what you need to understand is that the building block for peptidoglycan is not just made of sugar, but also a chain of amino acids, a short peptide chain of amino acid. And this structure is called NAG for N-acetylglucosamine and NAM for N-acetylmuramic acid. So you have NAG and NAM subunits, and they link together. See, here is a NAG-NAM disaccharide with the peptide bond. You see these dots? These are the amino acids that are attached to NAM. And these NAG-NAM subunits link to one another. So you have NAG, NAM, and you have long chains of NAG and NAM. And these parallel chains, you see this is a chain, here's a parallel chain, here's a parallel chain. Notice how the parallel chains link to one another. See, this NAM has a peptide chain which is cross-linked. This is called a peptidal bridge, or a tetrapeptide cross bridge, which means that there's a cross bridge, amino acid cross bridge between this NAM peptide and this NAM peptide. All right. So again, you have parallel chains of NAG-NAM subunits, and they're cross-linked with one another, connected by amino acids. You can see how this would make for a strong cell wall, right? So what's the difference between gram-positive architecture cell wall and gram-negative architecture cell wall? Let's talk about that. With gram-positive cell walls, you have a relatively thick layer of this peptidoglycan, these NAG-NAM repeats. And these also contain these unique chemicals called teichoic acids. So take a look. Here is a gram-positive bacterial cell. If we were to do a cross-section of its cell wall, like look inside of the cell wall, we would see this. And if you zoom in, what do you see? Here's the plasma membrane, and outside of that you have the cell wall. You see this thick layer here? This is very thick layer of peptidoglycan. And these structures, these tall structures inside, are called the lipoteichoic acids. Okay, the lipoteichoic acids, which help give a negative charge to the cell wall. Okay, these lipoteichoic acids help give a negative charge to the cell wall. The peptidoglycan is very thick. Okay, and this is what we know as the gram-positive architecture. So if you see a bacteria which has a plasma membrane and then a gap, and then a thick layer of peptidoglycan with teichoic acids inside. That's known as a gram-positive cell. Certain cells have gram-positive architecture. Certain species of bacteria have gram-positive architecture. Other species have gram-negative architecture. So what does that look like? What does that mean, gram-negative bacterial cell walls? Well, this one has a thin layer of peptidoglycan, okay? Not a thick layer, a thin layer of peptidoglycan. And not only that, you have two membranes, two cell membranes. There's actually a bilayer membrane outside of the peptidoglycan, which contains phospholipids, proteins, and an outer layer called the LPS, or lipopolysaccharide. The lipopolysaccharide contains a lipid portion called lipid A, and this is a very dangerous part of the cell. This is a toxin called endotoxin. We're going to talk about this in a subsequent chapter, but lipid A, which is again part of the LPS, which is part of the cell wall, lipid A is very poisonous, or I should say toxic to your system. It can cause fever, blood vessel dilation, inflammation, shock, blood clotting, even death. Okay, so let me show you what the gram-negative cell wall looks like. Here's a gram-negative cell. If we were to do a cutaway of the cell wall, this is what we would see. What do we have here? If we zoom in, we have a plasma membrane. Remember I said earlier, all cells have a plasma membrane. Okay, then we have here, you see here, this peach color structure, that's a thin layer of peptidoglycan. Compare that to this one. Look how thick the peptidoglycan layer was for a gram-positive cell. Look how relatively thin the peptidoglycan cell wall is in a gram-negative. And then look at here, what is this? This is a whole nother outer membrane. This is called the outer membrane. And the outer membrane has a blue layer. You see the bottom layer is blue. That's regular phospholipids. And the outer part of the outer layer is in this red color or orange color. What is this? This is your LPS, right? LPS is the outer part of the outer layer, LPS. And remember, there's a dangerous part to the LPS, which is called lipid A. Lipid A is the lipid part of the outer membrane of the outer membrane, and lipid A is called endotoxin. It's highly toxic to your system. There's also these porous protein structures in the outer membrane called porins. Porins allow lots of molecules through. So porins allow molecules to pass in and out of the outer membrane, and then you have your thin peptidoglycan layer, and then you have your inner membrane. So again, to review, gram-negative cells have a plasma membrane followed by a thin layer of peptidoglycan, followed by an outer membrane. And remember, the outer part of the outer membrane, this peach color, is called LPS. And LPS has a dangerous component called lipid A. Lipid A is the lipid portion of LPS. And lipid A is also known as endotoxin. It is very scary stuff. In your system, it can cause toxic shock syndrome. Now look at this. Compare that to gram-positive cells. Very simple. You have your plasma membrane, and then you have a very thick layer of peptidoglycan. You do not have an outer membrane. You just have a thick layer of peptidoglycan, and you have these teichoic acids. Okay, that's the main difference between gram-positive and gram-negative cells. And remember what I told you earlier? Not all bacteria have a cell wall. Some don't. And remember, especially the ones that don't have a particular shape. Those pleomorphic bacteria, the bacteria that don't have a particular shape, those ones don't have cell walls. Now, these also tend to be the small bacteria. Okay, the ones that are very small, on the smaller side of bacteria. Now, let's talk a little bit more about the structure of the cytoplasmic membrane, the cell membrane. Remember, it's known as a phospholipid bilayer. Each membrane in the cell is a phospholipid bilayer. And inside of that bilayer, you can have proteins that are floating around in the bilayer. It's a fluid mosaic model because it's fluid because the phospholipid bilayer is not static. It is not stuck in place. There's movement. The phospholipids are free to move about laterally. And then it's a mosaic of different proteins. So you have a myriad of different proteins that are interspersed. You see here, here's your phospholipid bilayer. Each one of these is a phospholipid. Remember, phospholipids are a type of lipid you learned about in Biology 1406. You've got the head region, which is hydrophilic, water-loving. And then you've got the tail regions, two tails, which are hydrophobic. And basically, a membrane is just two layers of these phospholipids. The heads point outside of the cell where there's fluid, water. And these heads point inside of the cell where there's also fluid, water. The tails point inwards. The tails point towards what's known as, you see this yellow zone? This yellow area is called the hydrophobic region, where there should be no water. There should be no water in this hydrophobic region. However, there is water outside of the cell. There is water inside of the cell. And that's why the hydrophilic heads can point out and in. Now look at these purple structures. These are called proteins. Okay, proteins. These proteins are floating in the plasma membrane inside of this bilayer. They're not stuck in place. You know why? Because each one of these phospholipids can move about, right? They're moving like an ocean. So these proteins are also moving around. They don't have to be stuck in place. That's why it's called a fluid mosaic model, a mosaic of all different kinds of proteins floating around in this fluid structure. Now, why do we have a cytoplasmic membrane? But why do bacteria have this membrane? Well, bacteria use their membranes for many things, not just to keep the inside of the bacteria inside and keep what's outside out. It's selectively permeable, which means it allows certain things in, certain things out. It protects the cell. Not only that, it can be used to produce ATP. You guys remember in Biology 1406, we discussed the mitochondria and how the mitochondria makes ATP by using the electron transport chain and all that, and ATP synthase. Do you recall? Well, bacteria have all of those components in the cell membrane, in the cell membrane itself. The plasma membrane harbors the electron transport chain and ATP synthase. So bacteria use their membranes for energy production to make ATP. It has the ATP synthase. It has the electron transport chain. It keeps what's inside inside. It keeps what's outside outside. It's selectively permeable. Again, it selects for what it allows in the cell, and it selects for what it allows out. Okay, it's very important stuff to understand. So look at this. By being selectively permeable, the cell can regulate the ion concentration inside of the cell. So what do cells do? They can maintain what's known as a cell potential. In this case, what's the cell doing? The cell is pumping positively charged ions, for example, sodium or potassium, right? Positively charged ions out, and negatively charged ions in, for example, chloride in. Notice what that would cause. That causes more negative charge ions inside of the cell, more positive charge outside of the cell, giving the cell a net negative charge at the cell membrane negative 70 millivolt to be exact, and this is called cell potential or membrane potential. And this is due to regulation of ions, right? Pumping ions in or out of the cell. Okay? By regulating ion pumping in and out of the cell maintains a negative net charge inside and a positive net charge outside. So let's get to passive transport. Do you guys remember how substances move across membranes? This was taught in Biology 1406, but we will reiterate. Remember, when you have a plasma membrane, only certain substances can cross the membrane directly without help or without a facilitator. Do you guys recall what those molecules are? Here's an example. Oxygen. Oxygen can cross the membrane without help. Oxygen can cross the membrane. Why can oxygen cross the membrane directly? What property does oxygen have? Oxygen is nonpolar. Does that make sense? Nonpolar small molecules can cross the membrane directly through a simple diffusion. Do you guys remember what diffusion means? Diffusion means a substance moves from an area of high concentration towards an area of low concentration. So which way would the oxygen molecules travel in this example? If they're following diffusion, which is a type of passive transport, which means no work has to be done. That's right, the oxygen molecules would flow into the cell across the membrane. What other molecules can cross the membrane directly that are small, nonpolar molecules? Well, what about CO2? CO2 can cross without help as well. What about methane, CH4? Methane, isn't that a small nonpolar molecule? Yes, methane, CH4, can also cross with simple diffusion. But what about these? What are these called? Do you see these little atoms, or I should say ions, because they have a charge. They're not called atoms, they're called ions. What about positively charged ions? Can they cross the membrane directly? Nope. Why not? Well, do you remember this yellow zone here? That's called the hydrophobic region. And charged things or polar things can't cross that zone. They bounce right off. So in this case, you need what's known as a facilitator, which is a protein that allows for these ions to cross. Does that make sense? This would be a protein, an integral protein in the cell membrane. This protein has a pore in the middle, like a channel or a carrier, and this allows for these ions to cross. So if these ions are crossing with just diffusion, which way would they cross? They would move from high concentration to low. Does that make sense? Across this facilitator. This is why it's called facilitated diffusion and not simple diffusion, by the way, because without this protein facilitator, the diffusion wouldn't occur. So when someone says facilitated diffusion, that means substance moving from high concentration to low through a facilitator, through a channel protein or a carrier protein. Does that make sense? When someone says simple diffusion across the membrane, they're talking about no protein required. The substance, because it's small and nonpolar, can cross the membrane directly from high concentration to low. All of these, by the way, anytime you're talking about diffusion, this is an example of passive transport. Now let's talk about facilitated diffusion, specific facilitated diffusion. This would be a carrier molecule. So let's say these molecules are polar. Well, there would be specific carriers that would attach to that molecule, letting it in through diffusion. Again, this would be a type of facilitated diffusion. And let's talk about osmosis. Osmosis means water diffusion across a membrane. That's what osmosis means, the diffusion of water across a membrane. Can water molecules cross the membrane directly like oxygen does? Think about that for a second. Can water molecules cross the membrane directly as oxygen can? The answer is no. Why? Because water molecules are polar polar molecules cannot cross the hydrophobic part of the membrane. So water molecules, if they want to cross into the cell or out of the cell, they must cross with a help of a facilitator. So diffusion of water, which is called osmosis, is a type of facilitated diffusion. Does that make sense? It's facilitated diffusion because without the help of this facilitator, this channel protein, and usually these are called aquaporins, by the way, the water molecules wouldn't be able to cross from high concentration to low. So speaking of diffusion or passive transport across the membrane, let's talk about how this occurs. A lot of this was covered in Biology 1406, but let's cover it again just so you're clear. Remember, small non-polar molecules can cross the membrane, the phospholipid bilayer, directly with no assistance, meaning that they cross without the help of a transporter. However, molecules that are polar or charged, so if you have ions, positively charged ions, negatively charged ions, if you have polar molecules, these molecules need to cross through a carrier. Okay, either a carrier or a channel. Remember the difference? A carrier interacts with the substance that's crossing and actually changes conformation to allow that substance in, whereas a channel just allows the substance to flow right across. Okay, so remember again, small nonpolar molecules cross the membrane directly through simple diffusion, whereas polar or charged molecules or water, which is a polar molecule, these require facilitators. They require facilitated diffusion. And do you remember the term for facilitated diffusion of water? That's right, it's called osmosis. Osmosis is the facilitated diffusion of water. So let's talk a little bit about how osmosis works. Remember, osmosis is facilitated diffusion of water. So let's say you have a membrane here. This is a membrane, a semi-permeable membrane, which means it allows only certain substance to cross. And let's say this membrane here, which I'm showing you with my pointer here, let's say this membrane is permeable to water, the blue stuff, but not to sugar, the pink dots. Okay, so which way would the water flow? Which way would osmosis occur? This is very important for you to understand. Before I explain it, let me remind you what diffusion means. What's the definition of diffusion? Ready? Diffusion is the movement of a substance, and that's spontaneous movement of a substance from high concentration to low. Okay, did you follow that? So a substance will diffuse from where that substance is high concentration to where that substance is low concentration. Now, you have to pay attention to what is doing the diffusion. In osmosis problems like this, what's moving? Remember, what's osmosis mean? Think about it. Osmosis is the diffusion of water across a membrane, right? Water is what's moving. So take a look at this. Where would you say the water concentration is higher? Look here. On the left side of this U-shaped tube, is the water concentration higher, or is it on the right side of this U-shaped tube? What do you think? What would you say? Where's the water concentration higher? Here on the left or here on the right? Think about it. Okay, you ready for the answer? The left. Okay, the water concentration is higher on the left. And I know what you're thinking. If you got it wrong, you're like, but Dr. D, there's more pink dots on the right. The concentration's higher on the right. Yes, the concentration of the pink dots is higher on the right. and these pink dots represent sugar, right? I told you these pink dots are sugar. So yes, the concentration of sugar is higher on the right. But I didn't ask you where the concentration of sugar is higher, did I? I asked you where the concentration of water is higher. And look here, there's more water anytime there's less sugar. Okay, I need you guys to understand this. The water concentration is always higher where the solute concentration is lower. Do you guys remember those terms? Here, look, solute. You know what solute means? Solute is the substance, in this case sugar, the substance that's dissolved in the solvent. What's the solvent in this case? Water, right? The solute is sugar, the solvent is water. So think about this. Water concentration's always going to be higher where solute concentration is lower. Does that make sense? So which way would the water want to diffuse? That's right, from higher water concentration to lower water concentration. So look what happens. Water flows to the right. Water net flows to the right. You know why? Because I told you this is a semi-permeable membrane that allows water but not solutes to cross. What if this membrane had bigger holes in it and it did allow sugar to cross, the pink dots to cross? What would you say would happen then? Well, if this membrane had big holes in it, yes. In that case, the solute, which is higher concentration on the right, the solute would cross to the left. Does that make sense? So the solute wants to diffuse to one side, to the left, whereas the solvent, or water, wants to dissolve towards the right. Did you guys follow all that? Hopefully you did. Hopefully that makes sense. Let me know in the comments below if it doesn't, because this can be tricky. Remember, the solute wants to diffuse the opposite way of the water, okay? So when I ask you osmosis problems, you're going to ask, which way is the water going? And actually these tonicity problems, do you see these? We're going to talk about these tonicity problems next. You see these? These are all tonicity problems. These are all essentially osmosis problems. I want you to know that all of these problems where I ask how, you know, sugar water or salt water, how the water concentration affects a cell, these are all osmosis problems, okay? So in all of these cases, you're going to ask yourself, which way is the water moving? Because the solute, which could be the sugar, the salt, what have you, the solute isn't crossing the cell membrane. It's the water that crosses the cell membrane. So every time you see these tonicity problems like this slide here, think about osmosis problems like this slide here. Okay, but the only difference is this semi-permeable membrane is the plasma membrane. It represents the plasma membrane. So let's look at these problems here. So on the top row, you have cells without a cell wall, such as animal cells or bacteria that don't have a cell wall. And on the bottom row, you have bacteria with a cell wall, or any cell with a cell wall, like plant cells have a cell wall. Okay, cell wall versus no cell wall. So let's see what happens. These are again called tonicity problems. You should have covered this in Biology 1406 already, but if you need a refresher, let's go ahead and do that. So this here, this first column represents an isotonic solution. You know what an isotonic solution is? It's a solution where the water and solute concentration outside matches the water and solute concentration inside. So the water and solute concentration outside meets the water and solute concentration inside. They match. In this case, for example, if I had an equal concentration of pink dots on both sides, would the water flow in any one direction? No. For every one water molecule that flows to the left, one water molecule would flow to the right. Does that make sense? So if the solute concentration outside matches the solute concentration inside, there's no reason for water to rush into the cell or a bunch of water to rush out of the cell. Does that make sense? Because remember, in these problems, what's moving across the membrane? The water. All of these are tonicity problems. All of these are osmosis problems. So an isotonic solution, does the cell gain water? No. Does the cell lose water? No. Does the cell remain unchanged? Yes. Same thing with the bacterial cell. Check this cell out, this bacterial cell. It's in an isotonic solution. Is it going to swell up? No. Is it going to shrink? No. The amount, for every one water molecule that enters the cell, one water molecule exits, okay? That's called an iso. Iso means equal tonic environment. Now, look at this column in the middle. This is what would happen to these cells in what's called a hypertonic solution. What does that mean? This is where it gets a little tricky, so I want you to bear with me, but hopefully you'll understand this by the time we're through. Hypertonic solution is where the solute concentration outside is far greater than the solute concentration inside the cell. So let's say I took a human cell and I placed it in high salt water, very high salt water. That would be the example of a hypertonic solution. So can the salt enter the cell? No, remember these are tonicity problems. In tonicity problems, the solute doesn't move across the membrane, does it? If the salt could enter the cell, would it? Yes, if the salt could enter the cell, the salt would go from where there's a high concentration of salt to where there's a low concentration of salt, so the salt would enter the cell, right? But because these are tonicity problems, which are essentially osmosis problems, you have to ask yourself, what's moving? Well, what's moving? Is the salt moving? No, remember, the salt's not moving. Only water's moving in these problems. So what is water doing when it's moving? Water is diffusing, right? Through a process called osmosis. And what does diffusion mean? Diffusion means the spontaneous movement of a substance from high concentration of itself, to low concentration of itself. So where is the water high concentration? In this case, where's the water high concentration? Inside the cell or outside the cell? I hope you said inside the cell, right? Because if you're surrounded by super salty water, well, then the water is more pure inside. The water is more concentrated inside. And the water is going to go from where water is high concentration inside to out, where it's less concentrated. So what does that result in? That results in a net efflux. That means net removal of water from the cell. And what does that result in? That's right, a shriveled, or they call it crenated cell. Now, what happens with the bacterial cell? Same thing. Water goes from where water is high concentration, leaves the cell to where water is low concentration. Okay? And the overall cell doesn't shrivel because the cell wall, which is made of peptidoglycan, the cell wall is very strong. But what does shrivel? Look at this blue. Look at this blue stuff that's getting all shriveled and wrinkly. Look at this blue line. That represents the plasma membrane. See, before the plasma membrane was nicely, tightly associated with the cell wall. But now, because all the water's leaving the cell, it's peeling away. Does that make sense? The plasma membrane peels away from the cell wall and becomes quite dehydrated inside. Okay? Now, let's take a look at the final example on the right. What happens if I take a human cell, for instance, and I place it in very pure water, like pure water? Where is the water more concentrated? Because remember, only the water can move. Well, if I place the cell in pure water, then that means outside of the cell is going to have more concentrated water, isn't it? So water is going to move from where water is high concentration into the cell where water is lower concentration. And isn't that going to cause my cell to swell? The cell swells until it can't take it anymore, and then it bursts like a balloon. Why does the cell burst like a balloon? Well, did it have a cell wall to keep it together? No, the plasma membrane can only take so much force, right? So when too much water is rushing into a cell and that cell doesn't have a cell wall, that cell becomes like a balloon that's ready to burst, right? That cell bursts. But if we have a cell wall around the cell, let's say this is a bacteria cell with the nice strong cell wall of peptidoglycan, or let's pretend this is a plant cell that has a cell wall of cellulose. In that case, water is still rushing in because this is a hypotonic environment or a hypotonic solution. That means the water concentration is really high outside and really low inside, so the water is rushing in. But does the cell pop like a balloon? Does it lyse? Remember lyse, L-Y-S-E? No, the cell does not lyse. Why? Because the cell wall keeps it all together. So bacteria that have an intact cell wall, they're fairly resistant to lysis in a hypotonic solution due to osmotic stress. Okay, this is another, when you hear the term osmotic stress, this is what they're talking about. They're talking about when water rushes into the cell and the cell becomes stressed to the point where it can lyse. Okay, so all of these were examples of passive transport, passive process. You know what passive means? Hopefully you remember that from Biology 1406. I just want to give you a quick refresher. Passive means that no energy is required. Okay, so another term for that is spontaneous or negative delta G. Do you guys remember the terms negative delta G, which means passive, which means spontaneous? Another term for it is exergonic. Okay, this means that the process is happening without energy input. Okay, diffusion and osmosis, facilitated diffusion, simple diffusion, osmosis, all of these are examples of passive energy, passive processes. Okay, because energy is not required. If anything, energy is released. Did you know that? Any passive process, energy is released. It's not required. However, you know, what if I wanted to take a substance like this, you see? What if I wanted to take a substance from where it's low concentration and push it out to where there's high concentration? Would that be following diffusion? Would that be passive? No, that would be active. That would require energy, wouldn't it? If I wanted to take a substance from where it's low concentration and push it out to where it's high concentration, that wouldn't be diffusion, would it? It would be going against diffusion. What they call that is against the concentration gradient. I'd be going against the concentration gradient. Whenever the cell wants to push a solute or some substance from low concentration to high, you're going against diffusion, you're going against the concentration gradient, and you're going to require energy. Okay? In that case, these are active processes. Active transport means energy is required. It's a positive delta G reaction. It's endergonic. It's non-spontaneous. Active transport. So a substance is moving from low concentration to high. So here's an example of active transport. These are all examples. Here a substance is moving from low concentration to high. See, ATP is required. ATP is used by the transporter to transport this substance from low concentration to high. Antiporter, same thing. You're using ATP to pump substances against the concentration gradient, in this case in both directions. And then you have coupled transport, where active transport of one substance results in active transport of a second substance through a separate transporter. Okay, and then you have group translocation as well. This just means when a substance crosses the membrane, it is modified in some way once it gets across. In this case, this sugar is crossing and the sugar is becoming phosphorylated, which means that it's gaining the addition of a phosphate group as it crosses the membrane. Okay, so that ends the discussion of transport across the membrane. Let's just continue a little bit more with some of the features of cells. Remember, the cytosol is the liquid portion of a cytoplasm. Sometimes we can use the terms cytosol and cytoplasm interchangeably, but don't get confused. Cytosol is the liquid portion of the cytoplasm. Cytoplasm is all the fluid inside of the cell. And this is mostly water, because most of your body weight is water. Okay, and don't forget, in a prokaryotic cell, the DNA is floating around in the cytosol as a nucleoid. And then you can have inclusions. Remember I said earlier inclusions exist in bacterial cells? And these are areas of storage. You could have phosphate inclusions or sulfur inclusions. These are little areas where deposits are made for storage. Now, let's talk about endospores. This is very interesting. Some bacteria can form what are known as endospore. And these are unique structures to bacteria. Not all bacteria, some bacteria. In fact, I'll tell you right now, only 2 genus of bacteria can form endospores. Bacteria in the genus Clostridium and bacteria in the genus Bacillus. We're going to learn a lot about this in subsequent chapters, but only 2 genus of the dozens or hundreds of genuses of bacteria, only 2 genus can form these endospores. So, what is an endospore? It's a defensive structure that's highly resistant against unfavorable conditions. So, unfavorable conditions meaning if it starts getting too hot, if it starts getting too salty, if it starts becoming bombarded with UV, if chemicals start coming in, if the cell is unhappy, and the cell meaning the vegetative cell is the normal bacterial cell. If the normal vegetative cell senses a poor condition, such as not enough nutrients, like I said, too much UV, too much heat, too much salt, too cold, whatever, the cell can transform into an endospore, where it basically forms this highly resistant spore. And this endospore can withstand horrible conditions, such as heat, radiation, chemicals. And that spore will germinate or grow out, right? Start growing back into a vegetative cell once it senses that those conditions have passed. So the heat's gone away, the radiation's gone away, the chemicals have gone away, or there's more nutrients in the environment. And here's how the endospore forms. You may want to know about this, like how the endospore forms. So first, remember the nucleoid, the circularized DNA, it gets copied in the vegetative cell. Now you have 2 copies of the nucleoid, you have 2 copies of the chromosome. Then the membrane, the cell membrane, kind of forms around both of these nuclei, or should I say nucleoids. And then what's known as the forespore forms. The forespore. The forespore is this this region that's going to become the spore. Okay, the membrane encloses around this nucleoid, and there what you form is two membranes around this copy of the DNA, and then the vegetative chromosome gets segmented and degraded. Now peptidoglycan goes into between the 2 membranes. That forms what's known as the cortex. And now a protein coat surrounds the spore, the developing spore, and that's called the spore coat. and then an outer spore coat forms, and then the endospore leaves the vegetative cell. The endospore can rupture the vegetative cell and travel on its own. So this is a spore. It's highly dehydrated as well. There's not much water in here. So drought conditions, like they call it desiccation, where water is removed. Lack of water cannot kill the spore. Like I said, radiation cannot kill this spore. This spore is like suspended in reality, right? It can't grow, but it also can't be killed unless you do some really radical things, which we'll talk about later, like autoclaving and things like that. Or using fire. Fire can kill the endospore. Autoclaving can kill the endospore, but not much else can kill an endospore. Not too many things. So these endospores are highly resistant, and once the endospore senses that the conditions have gotten better, the endospore can grow back into the vegetative cell, which means the normal growing cell. Okay, so this is really interesting stuff. Now let's just finish up by talking about some of the other parts of the cell. These are non-membranous organelles. Remember, ribosomes are found in all cells. Ribosomes are non-membranous. They are the site of protein synthesis. They are made of protein and RNA. Did you know that? Ribosomes are part protein and part RNA. The RNA part of the ribosome is called ribosomal RNA. Then you can have the cytoskeleton as well, which we're not going to talk about too much in this class. Remember, we talked about glycocalyces, flagella, Fimbriae. We're not going to cover hammy. Okay, and now we're talking about archaea a little bit. Sorry, we are talking about archaea here. Archaea have a lot of those similar structures to bacteria, but remember, they do not have peptidoglycan, right? Their cell walls do not have peptidoglycan, instead contain a variety of specialized polysaccharides and proteins. These S layers and things like that, we might touch on that later on. Obviously, all archaea have cytoplasmic membranes. All cells have a membrane. And that's about where I want to get with archaea. Don't worry about this slide here. All right, so that leads me to the end of that chapter. I hope that chapter 3 is largely review for you, but we did talk more about prokaryotic cells. Let me know in the comments below if you have any questions, or email me if you have any questions, and I'll be glad to answer them. Again, this is Dr. D, and I catch you guys next time.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.